Guys, we are discussing about RBCs. RBC's full form is red blood cell that constitute 99% of the blood cells. These are the smallest cells in the mammalian body. As we know, the sperm is the smallest cell in the body and the nerve cell is the largest cell in the body. So, the RBCs are in that between. Their function is the transportation of oxygen to the cell and tissue, carbon dioxide of the cells and tissue to the lungs for the exchange of O2. So we know that RBCs there is a hemoglobin which transport oxygen. So we are discussing the properties of erythrocyte, which means RBCs. Red blood cells or erythrocytes contain hemoglobin. Hemoglobin which contain iron. Hemoglobin give the red color of blood. Individual erythrocytes are orange, yellow, and in bunch they look like red. On configuration, erythrocytes are heavier and settle to the bottom to form a packed cell packet. White blood cells form a layer buffy coat on the top of the red blood cells pack, and platelets are settled on the top of white blood cells. Erythrocytes are non-motile cells but withstand deformation and passes through smallest capillaries and venules and remarkably elastic. So, erythrocytes can passes through the capillaries. Erythrocytes are circular biconcave disc in the most mammals but oval in camel. Erythrocytes shape which help them to carry oxygen with them. Mammalian erythrocytes have no nuclei, which is called anucleated. And there is important that camel having erythrocyte with nucleus. Lifespan of RBCs is 120 days only, short because of lack of nuclei, which contain genetic information, but that is not present in RBCs. RBCs have a semi-permeable trilaminar membrane. RBCs are elastic, alter shape to passes through capillaries. The properties of elasticity in RBCs help them to passes through capillaries and exchange of gas. Biconcave discoid shape maintained by membrane protein spectrin, which is present on the membrane of RBCs. Shape affected by osmotic force, which means osmotic force can affect or can burst RBCs. RBCs is short colloid to change shape in various sizes and vessels. Erythrocytes morphology. Morphology means how they look by outside. Mammalian erythrocytes are non-nucleated. Also we discussed in the previous part. And all other erythrocytes are nucleated. In the blood swimmers, RBC is stained with Giemsa stain and pink because they bind eosin, which is acidic. RBCs constitute 99% of blood cells. In blood, RBCs are found densely. RBCs are fairly round, 7.5 micrometer diameter. Thin central stain less than outer thicker annular area. RBC cytoplasmic content. Cytoplasmic content means what is present inside the RBCs. RBCs have certain protein and lipid. Proteins and lipids also present in the membrane called phospholipids. Delicate network granule is demonstrable in the reticulocytes and proerythrocytes. RBCs have hemoglobin, globin part, heme part and globin part. Globin is a protein and iron containing heme pigment is present which form hemoglobin which carry oxygen in the RBCs. Function of erythrocytes. Function of RBCs mainly contain to carry hemoglobin which carry oxygen. Transport of O2 to the cells, they reduce to Hb. Hb have a high affinity for oxygen. And in last part we are discussing about destruction of erythrocytes. Cell decrease demorphability in microcirculation is associated with increase in red cell rigidity, increase in blood viscosity and impeded blood flow can affect RBCs. Cell fragmentation is also affect RBCs.